So we can move on to the next presentation, which will be given by the Dr. Elsa Mazari. So she's a postdoctoral researcher at the St. Louis Hospital in Paris. So Dr. Mazari, you can share your screen.
First, thank you very much for the opportunity to talk about our work. And today we will show you how you can use PRIMO technology to reconstruct functional intrahepatic bile ducts with predefined geometry. But first, I would like to introduce you our team. So we are located at St. Louis Hospital in Paris. The team is led by Alexandra Fuchs and François Châtelain. And we see us as a multidisciplinary platform for tissue engineering. And as we are involved in different tissue engineering projects, let me introduce you the member of the team. So there was Wissam that was working with bioprinted esophagus in the project with Pierre Catanlab. We got also a PhD student named Yanis that is working on the cornea project. And there is also a PhD student named Sarah that is working on gut organoids. Today, I will talk about more my project with Dimitri that is focusing on liver project. So let's start with the liver project. When you are thinking about liver tissue engineering, a lot of studies are focusing on reconstructing the parenchymal tissue with hepatocytes. It is really, really important, of course, but there is another system that is really important, essential in the liver. It's the biliary tree. The biliary tree may be found in all the liver at different scale. It's really important to actually transport the bile from the hepatocyte and to drain it out from the liver because the bile is very toxic for the hepatocytes. And this system is constituted by cholangiocytes that are really a big transport system that is going all along the liver at different scales. So there is large bile duct and there is small bile duct. And here is the problem in liver tissue engineering. There is not so much study to reconstitute such a small duct. If we are going inside the liver lobe, that is a small liver unit, very small one, the cross section that you see, you can see the small bile ductules that are around 10 and 100 micron. The size is really tiny, as you can see, but the geometry is also very complex. And there is a real challenge here to reproduce in vitro such part of the biliary tree. And here is our work. Actually, we want to use the PRIMO to reconstruct such intrahepatic biliary tree. So for the methodology, what we are doing, we are using a PRIMO system for the microfabrication. Actually, we select different natural matrices that are photopolymerizable, and we mix it with the cells, the cholangiocyte, the cells that are responsible for the bile duct system, and we put finally, what we want to do is to prove that we got, after culture, biliary cells that are able to organize and to be functional within tubular structure that are at the intrahepatic scale. So I will spare you the detail today about how the microfabrication with PRIMO can occur. The main thing here for us is we use it as a tool to provide us the control size and geometry of our structure, still under 100 micrometer in size. So just a take-home message, we here want to build a relevant cellular microenvironment using PRIMO to help to guide cholangiocyte into branch network at the intrahepatic scale. So when I say that, I say almost everything. So the presentation today will be very simple. There is something else that I need to add, and it will be really complete. It concerns the general methods. We saw different application of PRIMO system today, a very interesting one. So as you understand, we use the PRIMO system a little bit different way. We use it as how to say, a system to encapsulate cells with controlled size and geometry. So the protocol to use it is quite a little bit different. So here is really what happened in the lab. You can see actually the device that we load with the photopolymerizable hydrogel and the cells on the left on the picture. We upload template with the relevant size and geometry that we want. And on the image on the right, what you can see is during photopolymerization process, you can see that the cells that are really round are trapped inside the photopolymerized structures that start to appear. After this, there is a cleaning step. So basically what we are doing, we are putting the structure in a culture medium, and the unphotopolymerized part of the matrices will be removed. And only what we say on the glass substrate will be the structure with the cells and the photopolymerized structure. So once you did that, you have your protocol, and the main complex thing is all the encapsulation parameters that need to be taken into account. So we try to address a list here that is not a complete list. You can add many, many other parameters, but just to let you know that, for example, for cell types here, it will be very simple because we will use only one cell type that is normal rat cholangiocyte. It's a cell line derived from the rat that are cholangiocyte. But after, for this system, you need to each time optimize, find the good condition concerning the composition of hydrogel, because different hydrogel are available. You have also to make a lot of experiment to find the good photopolymerization condition, especially regarding the UV dose, because cells will maybe have a poor viability if you are using too high UV dose. You have also to check for the fabrication condition. Initial cell density is very important, and the size of the pattern and the geometry that you will implement will be also very important. And finally, you have to check for the culture condition of your 3D structure after. The culture medium, is it optimal for 3D system? Do you use growth factor? Will you use flow rate? So here is a big picture of such experiment. Now I will show you successful results when you address all these issues. But just to let you know that if you want to use PRIMO to encapsulate cells, it's really definitely possible, but you need to have an intensive time of preliminary experiment and try hard, but be confident it will work after. So about the main results. So first, thanks to bibliography regarding cholangiocyte, we found one mix of photopolymerizable matrices that is made with collagen, acid hyaluronic, and fibrinogen, and we mix our cells, our cholangiocytes, with this kind of mixture and photopolymerize it regarding different patterns. So here you have the pattern with the trees and the pattern with the line of 100 micron and 50 micron. Just immediately after fabrication, as you can see, the cells are trapped in the structure but stay very round. And all the structures that were not polymerized were removed. But what is very interesting, it's just 2 days after fabrication, the cells were already able to migrate, to proliferate, and to start to form structure within the photopolymerized structure. So it was a good answer from the system that our mix will be okay. But as I said, we wait for 1 week of culture to see how the cells auto-organize into this tubular structure. So to characterize this mixture with collagen, hyaluronic acid, and fibrinogen, what we did also is to see if we remove one or two components of this mixture, what will occur over time culture. And as you can see in this table, actually, if you remove one or two components from this matrix, after 1 week of culture, the formation of tubular structure will fail. Except for our successful mix with collagen, hyaluronic acid, and fibrinogen. Another thing that we characterize is as we can play with UV doses received by a photopolymerized structure, we characterize the properties of the mechanical properties of the gel after photopolymerization. And as expected, when you double the dose, you have more rigid substrate, as you can see on the graph on the top. But unfortunately, when you have more rigid substrate, what happens is the formation of the tubular structure fail after 7 days of culture, as you can see with the image. And at the same time, also as expected, we check for the cell viability compared to UV doses. And when the UV doses is higher, the viability of cells decrease really rapidly. So this experiment shows us the encapsulation, here again, the encapsulation parameters that we should use for our experiment. We say that the mix that we should use is made with collagen, hyaluronic acid, fibrinogen, with a UV dose of 20 millijoule per millimeter square. That is really, really small UV dose compared to other applications, as I saw today. Maximum 20 millijoule per millimeter square, and this lead to the successful formation of tubular structure after 7 days. One other key component of our system is the use of Matrigel in the culture medium during culture time. We saw that if you do not have Matrigel in the medium of our tubular structure, the tubular structure are not stable over time culture, as you can see in those pictures. Actually, what we think is we hypothesize that the Matrigel is mainly made with laminin, and the laminin is really important signal for the polarization of the cells over culture time. So Matrigel is required for our culture, but once you put Matrigel in the culture system, you can have stable tube formation over 21 days. After this, what we did is to characterize our structure, biliary structure, using confocal microscopy. So first, what you can see in A and B, it's two different lines with total diameter for the first one of 20 micron and the second one of 50 micron. And you can see that the luminal space seems to be first closed and empty, with a luminal space of, let's say, 15 micron for the first one and 40 micron for the second one. What you can see also is the F-actin distribution that is characteristic of epithelial cells with dense band along cell-cell border. You can also check for the Z01 staining, that is the staining of tight junction, and we can see that the cells, the tube that are formed seems to form an intact epithelial barrier. And finally, on C, what you can see is the staining of the acetylate alpha-tubulin, that is the main component of primary cilia. And we are very happy to see that the cells were well polymerized with the primary cilia facing the lumen, as you can see in those pictures. Finally, as I said at the beginning, we want to characterize the functionality of our tubular structure after culture. So we employ two different regular functional tests. The first one is named FDA test for fluorescent diacetate assay. So this component is not fluorescent at the beginning, but biliary cells are able to internalize it, to transform it into fluorescein, and to transport it actively toward apical side of the membrane and put it in and disperse it in the lumen of the structure, showing their functionality. So it what you can see on the left panel of the slide, and this transport of fluorescein is involved by a receptor named MRP2. And the other test that we employ is tests named rhodamine 1 to 3 that employ another apical receptor named MGR1. And we can see also that this fluorescent component is actively transported from the outside, from the culture medium to the lumen of the structure after 7 days of culture. So for us, it was a successful formation of intrahepatic bile ducts. So we published all our results and more in this publication. Do not hesitate to go and see it if you want to know more, or if you want to cite us. You are very welcome. But I do not want to end with this. I want to address all the many questions that we still got, and we are still working with this concerning our system. So, as I said, we construct these bile ducts for liver tissue engineering, and we are really looking forward to knowing what happens if we mix our bile ducts with hepatocytes or hepatic tissue. So many questions here. What cell culture should we use? What kind of hepatic tissue should we use? Spheroid or cell sheet? And how do we image the micro-size Ewing canal that are really deep in the fabricated tissue? It's really many technological and open questions. Another thing that we are still working on it, and we are really interested in this, actually, I said that the biliary tree is responsible for the transport of the bile. So is in our stable tube, if there is any spontaneous flow now, can we characterize it? We are really interested in this because actually it also involves the fact that we need to connect our structure to outside. So we are still struggling with this. And of course, if there is anything secreted in our structure, can we recover it? And can we recover the structure? So many open questions. I just give you a few really quick answer about how we can address this problem. So first, we have a collaboration with Mireille Chevalier in Sierra Grenoble, that is an expert of using micro-patterning, glass slide, making spheroid of hepatocytes. So we are really eager to put a spheroid in our structure. So still the problem of culture medium. So if anyone has any idea about this, they are really welcome. Another idea, as I said, we are really excited about this, is connecting our structure to the outside. So the first idea we got is to take microcapillary and directly connect them during photopolymerization, as you can see on the picture, and see what happens. So hopefully the capillaries stay, as you can see, and on the bottom you can see the same results with cells that are endothelial cells, and here is a film where you will be able to see how the cells are proliferating and migrating around the tube. But the good thing is the tube stays, And the cells seems to be not so perturbed by the presence of the our epithelial cells, our biliary cells. So the cells seems to be able to, after 7 days of culture, to be organized around the tube. So just have to connect it to the pump now to see what happened. And also I told you about we really would like to recover our structure from the glass substrate, because actually it's not possible to take this structure and to transplant this structure because there are culture on the top of the glass substrate. So the idea here is to recover all the structure using an arginate patch. So what we are basically doing is we put arginate and gelate it on the top of the structure, and the arginate take all the structure from the detaching all the structure from the glass substrate. And you can see after 7 days of culture in arginate a tree that seems to be intact. So we are still developing this way to recover our structure after photopolymerization and maturation in the incubator. But it will be a wonderful tool to perform transplantation or even co-culture system like this. So at the end, I would like to thank, of course, my team mate and cell therapy human research team, Cytomorpholab team, and also Alvéole for the invitation. And thank you for your attention.
Thank you very much, Dr. Mazari. So I have a question. So you were in the last slide talking about the encourage, recovering of the hydrogel. So I was also wondering, do you have to treat your glass before polymerizing the hydrogel to make it adhere, let's say, to the glass, or there is no particular protocol to do that? It attaches by itself.
Yeah, thank you for the very nice question. You need to treat the glass before, because we are depositing thick gel of around 100 micron to 200 micron. So you need to put some siliconized coating at the beginning before your experiment.
Okay, thanks.
Siliconization.
Yeah. And also, still concerning the stability of hydrogel, let's say. So I know that some people struggle to culture cells embedded in hydrogels because actually they contract and they shrink the gel. Is it something that that happened with your system or not?
This is something that we are really searching for our system, because we want the cells remodel totally the gel. And make what they want with the gel Actually, what we are doing is providing a relevant cellular microenvironment, so the cells can behave like they are doing normally in vivo. So we want the cells to remodel totally to get rid of the gel. For example, for this application, we do not want to have we want to have lumens that are totally empty. So the cells are able to drain out totally the initial hydrogel, and we are looking for this. We are looking for the cells that are able to remodel and change the gel.
Okay, thanks. And we have a question from Benoît Ladou. So it's just wondering about cell death due to UVs.
Yeah, so that's why we characterize just after photopolymerization. We mean it by 4 hours after and 1 day after. So what we can say about this, it's here. It's of course, the higher the dose is, the cell death will increase a lot. But you can minimize the UV dose received by the cells because we are working with, as you can see, a really low dose from 10 millijoule per millimeter square to 20. But you can also add some things in your mix to avoid also the UV to be so damageful to yourself. From my experience, I use different cell types. There is also photosensibility, different cell photosensibility, but I never found some cell types that are not able to be photopolymerized in condition with low dose.
Okay, thank you very much, Dr. Mazari, for this nice presentation. We know that it's always a big challenge to do a 3D cell culture with hydrogel polymerization, so thank you very much.